Yep, I'm still here.
Good. Haven't lost you. That's great. All right. All right. We are live now. Let me see how many. We have 90 attendees. There were only 80 when I spoke to you last, but there are now 90. There's a rush of last-minute registrations.
Great.
Yeah. But at the moment, there's just you and I in the room. No, somebody has just come in.
They're probably all checking once every few minutes and then
Exactly.
we'll be on by the time we actually start.
So we have Lindsay, we have Andre, and we have Belinda at the moment. Let me see. Perhaps, guys, if you can hear us, just maybe type a little yes in the chat box. Make sure we're getting through to everybody. The chat box is on the right-hand side of your screen, guys. So we'll give it a few minutes, Elaine.
Okay. Will people be able to write in questions in the chat box?
Yeah. Yes, for sure. Can you see that chat? You can see that chat box on your right-hand side of your screen. Can you see that at your end?
Okay, so if I reduce the size of the screen, does that mess up the slides on the screen at all?
No, it shouldn't. It shouldn't do, but
yeah. So I'm asking you that when you see that what you're seeing in that main box, are you?
Yeah, there should be a box on the, but it doesn't matter. I mean, I can repeat the question, so that's no big deal. So
yeah,
should be okay.
So what you might want to be doing is during the course of my lecture is to have like groupings of questions, because people will ask pretty much the same thing over and over. And so if you could have maybe 5 questions to start out the question and answer, culled from what they put into the box.
Sure, no drama. Okay, so let me see. We have 13 people at the moment have logged in. Good morning, everybody. Well, good morning from Australia. Anyway, I think most of our attendees are from Australia, Elaine. So I think we have a couple that have registered from, I think, France and Germany.
Okay. All right.
But that's all good. Probably should have opened the room up probably another 5 minutes earlier.
Yeah.
For everybody to come in. But we have 13 people in the room at the moment.
And they'll roll in a few more minutes, so we'll get started in and maybe right on the dot at 3:35 or something like that.
Yeah, what time is it there at the moment? It would be 3.
It's actually, it depends on which clock I look at. According to my computer, it's 2:31 p.m. here.
Okay, all right. So yeah, give it till 2:35, and then we'll go. But then again, people can see the replay anyway. So
great. Okay. So if they miss 5 or 10 minutes, then they can come back and
see the replay. Yeah. All right. Well, good morning, Shara. Shara is another attendee who's just typed in good morning.
There's Michelle.
Okay, Michelle. And she's also off of YouTube as well, which is interesting. This is a little bit of a glitch we found with this software, is that some people have to go to the YouTube channel, which is the SFI Australia YouTube channel, to actually see this live streaming. And I don't understand why, but I might speak to the webinar software people about that. It happens on some, but not all of the webinars, and I really don't know why. Anyway, okay, let me see. How many do we have now? We have 13 still. But I think I'll formally start, if you like, Elaine.
Okay.
Good morning, everybody. My name is David Ellery from Soil Food Web Institute, Australia. And thanks for attending our live webinar Q&A session with Dr. Elaine Ingham. Just for a bit of background for those who don't know, I think a lot of people that are attending today have either dealt with Christopher or myself in the past, but for those that haven't, Soil Food Web Institute was actually started by Elaine. When was it, Elaine? Back in 2000? 2000?
Yeah, 2000.
Yep.
Right
Yep.
before, right before the turn of the century. So we actually got up and got going in 1999, but kind of opened our doors for most samples coming in late in 1999.
So then, things have been bubbling along since then, and Elaine has become extremely busy with her work over in the US. So my brother Christopher and I basically took over the reins of SFI. Still work very closely with Elaine. But our background, if you like, my brother's and my background is in organic soil amendments and has been since, again, 1999, which is when we first met Elaine. I think we met at the Millennium in Michigan, I think it was.
Yes. Oh,
Remember that?
goodness. Yes,
Yeah.
I remember that. A few
So
years ago.
exactly. So we both attended that, and Elaine was a speaker at that, and so was I in the area of organic waste and organic waste management. So there's a little bit of history for everybody. Maybe I'll ask Elaine to say a few words, and then we'll get into this morning's webinar.
Well, I'm very glad that you invited me to come and give this talk. I hope to be able to come to Australia later this year, probably sometime. I think August might be an opportune time to go over the middle of your winter, middle of our summer, and so I'll miss the really hot, disgusting days of August here in the U.S. So hopefully we'll be able to get that trip together, and I'll be able to do some work while I'm over there in Australia. The things have been really moving here, around the world. I've gotten to the point where I really can't handle all of the work, the interactions that I'm trying to do with people. So have joined forces with another group called Environment Celebration. And when I was working at the Rodale Institute as the chief scientist there, I met a group of funders who wanted to know what had to be done to move along the scientific acceptance of the work that I've been doing. And I, of course, said, well, having plots where I can maintain control, where I can do research on those plots year after year. When we're trying to publish this kind of work in agriculture, we have to maintain the plots for at least 3 years in order to have 3 years worth of data. And you have to have multiple treatments of the same thing, at least 3 of them in your fields. 5 is better. Well, 100 would be better, but you have to get practical at a certain point too. And so I said that's what I really needed, was a place where I could maintain the treatments, because if I'm working with a farmer, we go to the farmer's place and we show them how to produce the sets of microorganisms that they need to replenish to put back into the soil because they've been killed by the toxic chemicals, the pesticides, the inorganic fertilizers, the too much tillage, things like that. They're not present in that dirt. And so we've got to convert that dirt back into soil. And you've got to understand how to grow those organisms and maintain them in your soil. And most growers, if they catch on, if they follow directions, they can see how much easier growing their crops are. And so within the first 6 months, they'll convert the whole farm and then say goodbye to the scientific experiments that I have to be able to document for 3 years, because the grower's purpose is not to do scientific research. Their goal is to make money so the farm will stay alive. So you can send your kids to college, you can put in that swimming pool, you can, whatever it is that you need to spend money on, you can do that. And then those people who can't follow directions and don't do it right, they just can't comprehend why measuring temperature and why turning the piles and why you have to apply things, remembering every second along the way that you're dealing with living organisms. These are like your children. These are your livestock, and they do so much work for you. You have to nurture them. You have to take care of them. And if they can't manage to do that, then they fail. And of course, their response is going to be, well, this biological stuff doesn't work. So whenever you hear somebody put on that attitude, you want to re work that verbiage that you just heard and say, okay, it's because they couldn't follow directions and they failed. Not that you can't do this with biology. So having faced that sort of situation for the 20 years since I left university and started working very closely with growers around the world, finally these funders came and said, what do you need? And that's what I needed. I needed a place where I can put in long-term research plots. And so in 2015, they bought a farm. And we started the Environment Celebration Institute in California. It's just north of Oroville in California. So we're up about 3,600 feet. Not down on the valley floor, thank goodness, because right now the valley floor is completely flooded. I don't know if in Australia you have been getting a lot of news from the U.S., but the United States tallest dam is in Oroville, Lake Oroville, and it's very likely, it is so close to failing, and that means so much of the valley north of Sacramento is going to be completely flooded. So it's kind of a grim situation. Luckily, we're above the dam and we don't have to worry about it. But it does make it interesting. All of our employees were staying at the farm with us because their homes were down in the town of Oroville and it was completely evacuated. So a little bit stressful times. But we've been there at the farm now for 2 years. And later in my talk today, I will be going over some of the data that we are publishing. For those of you who might want to be able to see it, go find the Permaculture magazine, and you can read the data that's coming out of our work. 2 years worth of data so far. And of course, we have to wait to publish because this year will be our 3rd year. It's no longer next year, it's this year. How time flies when you're having fun. So yeah, lots of interesting things going on. We have online courses, and I think, Chris, you've pointed people to those online courses. And so if people are interested in what I'm talking about today, you probably want to go a little bit more in depth, get a lot more practical, hands-on, when and how and why do I do these things. And as a scientist, I always have to explain why things work. This is not magic. We can repeat what we're doing time after time after time after time, but you do have to do the proper management. I do like to kind of liken developing these proper sets of microorganisms to go back on your soil as a lot like learning to make an angel food cake. If you don't follow the directions, if you don't have the proper starting materials, you are going to have complete and total catastrophic failure. You cannot make an angel food cake if you don't pay attention to the proper methodology. And exactly the same thing happens with biology. If you don't understand what you're doing, then you're not going to be successful at doing this. So, did you want to talk at all about the courses that you guys are offering?
Yeah, during the webinar and also later on, guys, there'll be links that I'll send to you, which for people that are interested in taking what Elaine's talking about much further, learning some of the intricacies of the subjects that Elaine's talking about. There are a series of online courses which is run by Soil Celebrations, and they're available through SFI Australia here at a heavily discounted rate. So I'll be interjecting bits and pieces during the webinar that people can see on their screens, and we'll take it from there. So I'll hand it back to you, Elaine.
Okay. Well, I think we'll get into today's presentation. So hopefully up on your screen is the first slide. It's the Soil Food Web. This is from the Lexicon of Sustainability. It's a group in the United States that puts together a calendar. And so 12 different people every year are highlighted on their calendars. And they go and interview, they give a summary overview of what the people that are leaders in the world of sustainability are doing. I know they've tapped quite a few people from Australia in the Lexicon of Sustainability calendars. As I understand it, I just happened to be the very first person on January of the very first year that they started putting out the calendars about people doing really important sustainable work all over the world. So I feel like I'm kind of the calendar girl for the Lexicon of Sustainability because I was first. So this gives a good overview of what I do. So you will see this food web picture a little bit later on in the seminar. So we are going to be starting with just the barest introduction to the topic of life in the soil here today. I do want to point out that I have all the alphabet soup after my name. Oh, aren't you impressed? And yeah, I've been through the academic mill. You have to be able to understand and comprehend the current scientific understanding in order to advance the frontiers of science. And so I've definitely done that in my life. Soil Food Web is the company that I started in 1996. And of course, then in the year 1999, headed over to Australia to establish a lab in Lismore. And David and Chris are now continuing with that lab and doing work with people over there in Australia. I do want to point out Environment Celebration Institute, which is the farm where we do the research. And anytime you're over in California and you want to come see the farm, please let us know that you want to come up. We give tours, but we like to have set tour times because there's just so much work to do on the farm. We have 60 acres and ever expanding the work that we're doing. The Environment Celebration Institute also handles the online courses in the US, but here in Australia, it's Soil Food Web Australia. So that's, make sure you contact the right entity, please. Because if you contact us in the US and we see that you have an Australian address, we'll send you to David and Chris. So the local people helping you get online and get things dealt with. We just recently have started working with Biodynamy, which is a large international group of people that are putting together management teams all over the globe so that we will have people that can come to folks' farm, be there for a growing season or a year or two, train your folks, get you fully trained and fully understanding the nuances of what we're trying to do here, which is to get everybody off those toxic chemicals, stop applying all these things that kill all of the organisms. Every single inorganic fertilizer is harming the biology in your soil, and especially at the concentrations that we're putting on urea or phosphate fertilizers or iron or calcium, or people always think of lime and gypsum as being organically okay. Not at all. Those are salts, and they will take water away from your microorganisms. And what I often like to say to people is, what would happen to you if we pushed you into a vat of salt? any one of the thousands of different kinds of salt. It's not just table salt. Um, what would happen to you if we pushed you into that vat of salt? All the water in your body would be sucked out of your body. You'd churn into a giant prune and die. And that's exactly what we do to organisms when we're putting on these inorganic chemical applications in our soil. So I don't care whether they call it a soil conditioner or they call it an inorganic fertilizer, whatever. You're destroying the very organisms that are the basis of life on this planet. For us to grow our food, we need those organisms. So we need to get off of those particular drugs. We have to exit those addictions. And it's always fun when you're fighting with corporations that are making billions of dollars a year. They, of course, do not want anybody to learn about this because you would probably be very upset if you understood just how much damage has been done. Because once you learn all of the things that these organisms do for you in the soil, you'll realize that this is the reason we don't have clean water. It's why all of these chemicals are moving from the land out into the barrier reef around Australia and killing that barrier reef. And what happens to Australia as a continent once the barrier reef is gone?
Mm.
Australia is going to be gone.
For sure.
So, yeah. So, if you would like to see your continent remain intact, you have to do something about this. And that means stop allowing all of these soluble inorganic nutrients to wash out of soil and into the water and destroy the water quality, destroy the life in that water. And of course, that water then comes back onto the land. We pump it out of the rivers and the lakes and streams, and we put it back on our agricultural fields and cause more damage. Every single pesticide is going to kill more of the beneficial organisms in your soil than it's going to kill of any of the target organisms. There's way more damage to your ecosystems, to your soil, than there is benefit by killing off a certain number of the pests or the problem organisms. And of course, the problem organisms get resistant to those toxic chemicals pretty rapidly. So we just have had to develop nastier and stronger and more toxic chemicals to try to deal with the problem. Well, that's not the way Mother Nature has ever dealt with holding these pest and disease organisms in control. That is not the way she does it. And we need to learn to mimic what nature does. So we stop fighting with Mother Nature. Because, of course, if we're declaring war, and we really have, as a species, human beings have declared war on nature. And in a war between nature and humans, ultimately, who's going to win?
Mm-hmm.
It's not human beings. So, yeah, maybe human beings have won the first few skirmishes. We have been able to control some diseases and pests, but not completely. And they come back nastier and stronger and more resistant to all the toxic chemicals. So how does nature do control on these organisms that are problems? And so that's part of what we're going to delve into today. So biodynamy, I was just trying to give a little blurb about that. So when you see that we've joined forces with biodynamy, it's because I need help with the business end of things. This is just getting too large. There's too many projects all over the world that I'm involved in and training people. And there's just not enough of me. So that's another group that I have, lent my expertise to, and they're going out and doing the big projects. So a little bit more background on myself. I think you can read this list of where I've been in my life, undergraduate school in Minnesota, double major in biology and chemistry. So I have a good background in chemistry as well as biology. Did my master's degree in marine microbiology, where I first ran into this problem of the methodologies we were using in microbiology to go out and assess biology in the real world, out in the environment, in soils, in sediment, on plant surfaces, on surfaces of human beings, were completely and absolutely just ridiculous. They did not measure what they thought they were measuring. They were using methods that were used in medical microbiology that were developed by Louis Pasteur and Robert Koch back at the beginning of modern medicine. And they were using those methods to try assess biology in the soil. Well, just about every organism in the soil cannot grow on plate counts. You cannot grow them in petri dishes in laboratories with the media that we were using, completely inappropriate for assessing that problem. I worked with microorganisms in the digestive tracts of oysters. And what I could see is all these hundreds of different media that I was trying in the laboratory to get things to grow from the digestive system of these oysters were growing one or two species, and that was it. Whereas I looked through my microscope at a drop of the digestive fluid from inside an oyster, and there were a million different species of bacteria and fungi, protozoa, and even nematodes inside those digestive systems of those oysters. We were missing just about everything. So if you go back to the scientific literature and you start reading papers from before 1985, just the methodology was completely wrong. Don't even bother reading them, because most of them are using methodology that's completely outdated, completely inappropriate. So learning that when I was working in marine microbiology at Texas A&M, from there I went to Colorado State University for my PhD, working on methods for assessing life in the soil. And so develop those methodologies using microscope approaches. And really, if you come to understand all the different ways that people will try to assess biology in the soil, there are always problems with each and every one. You have to understand the method in order to interpret the information properly. So if you're doing enzyme tests, if you're doing substrate utilization, if you're doing chloroform fumigations, I'm sorry, folks, you're missing most of everything. Trying to use those methods to tell you what's going on in soil. Learn how to use a microscope, and we do that. I know that Chris is going to be doing a microscope class here not too far into the future. So you might be interested, if you can't make the next class, there's going to be another one. And so we point you to very inexpensive microscopes. They cost $300, and they come with cameras, so you can take pictures. And when you can't identify something, you can send those pictures to me or Chris, and we will help you figure out what it is you're looking at. So come take the course, take the online course. And then, of course, it's always useful to have some hands-on, come bring your microscopes and get a little bit of practical hands-on training. Bring your own soils so you can take a look.
For everybody's benefit, the courses that Elaine's talking about are being held in Lismore around about between the 23rd and the 28th of February. So it's this month. If people want to know, then know more about it, then just go to either the Living Soils website, where most people registered for this webinar, or through to the SFI website, and just send us an email. Sorry about that, Elaine. Over to you.
Yep, thanks. Yeah, and when you need to interject something, you probably just want to say my name, and I'll know that you're really mean to catch my attention. Okay.
No problem.
So University of Georgia, Oregon State, where I was a professor for about 15 years. And because I had a graduate student that worked on genetically engineered bacteria and showed that, in fact, genetically engineered bacteria can have absolutely unpredicted consequences. And if we had actually released that particular bacterium out into the real world, we would have lost terrestrial plants. That might impact your life just a little bit, right? No more corn, no more tomatoes, no more parsley, no more carrots, no more apples, no more bananas, no more terrestrial plants. Because that genetically engineered microorganism now had the ability to produce alcohol in the root systems of plants, using the exudates that the plants themselves were putting out of their root system in order to grow microorganisms in the soil, Um, and so, presented that information at the United Nations, and of course, Monsanto went crazy. So I was no longer a professor at Oregon State University. I started Soil Food Web in 1996, and it continues on to the present. I did about a 3-year stint at the Rodale Institute in Pennsylvania as their chief scientist, and then came back to Soil Food Web and am continuing to do research. As I said, we have our own research farm now, and I'll be talking about some of those results. So why be concerned with soil life? Just in case you don't really have a grasp on that yet, healthy plants don't need toxic chemicals in order to grow, and we'll go through the reasoning and show you why it's not necessary. Healthy plants have the proper balance of nutrients so that the produce that we're making has a good flavor. It satisfies your hunger very rapidly. You don't have to eat as much because you're not eating empty calories. You're eating the nutrients that your body requires. We've been doing a fair amount of work with people like David Johnson at the University of Mexico, with Rodney Rodriguez at the University of Cornell, and the human microbiome issue. And they've been showing all these hundreds of thousands, if not millions, of bacteria and fungi, protozoa and nematodes that have to exist in your digestive system in order for you to make nutrients available to yourself. And if you lack these microorganisms, you're not going to have proper digestive system function. And that affects every organ in your body. When we start looking at some of these diseases that have gotten worse and worse and worse over a very short period of time, from the time I was a teenager to now, these diseases, these problems have increased by 300%, 800%, just massive amounts. It's frightening. And they are linking that to destroying the biology in your digestive system. So how do you replenish the biology in your digestive system? You have to be eating plants that have the right sets of microorganisms on their surfaces. You got to make sure they're the good guys, not the bad guys. And where do those organisms on your plant surfaces come from? When you eat a carrot, that's the root of a carrot plant, and you need to have the right soil organisms on the surfaces of the carrots to re-inoculate the correct sets of microorganisms in your microbiome. And so when you're eating sterilized food that you buy at a grocery store, you're not getting replenishment of those organisms that you require. When you eat an apple, you should be eating a whole herd of these really beneficial microorganisms that should be living on the surface of your apple. And that replenishes the biology in your digestive system. But if we're sterilizing our food and we're sterilizing our soil, we're killing all the beneficials in our soils off and turning our soils into dirt. And all of those nutrients and all those toxics are coming out of our soil and going into our drinking water. No wonder we have type 2 diabetes. No wonder that we have all these arthritis and rheumatoid problems and inflammations, and directly connected, being shown beyond a shadow of a doubt by the medical community that without the right biology in your digestive system, you're going to suffer all these problems. So if you want to age gracefully and not as painful, you have to have the right sets of organisms on the food that you're eating. And that means they have to be in the soil. So flavor, reestablishing your digestive system health all starts with the soil. We have to stop destroying our soil. So when we start thinking about plants and nutrient concentration to give you flavor in the foods that you're eating and to give you the nutrition that you require, where do plants get their nutrients from? All but two nutrients come from the soil. So what are the two things that your plant is going to be getting from above ground? And people usually have to sit back and really think about that. And because a lot of times people will say, well, water comes from above ground. But most of the water that your plant gets has to come through the root system. So it comes through the soil. It's cleaned up, it is altered, it is made into what the plant requires with all the proper nutrients in it through the soil. And so exactly what are these processes? What's going on in the soil? What do we need to understand? Because we've got to reestablish that normal nutrient cycling process so that we can have healthy plants, so that we can be healthy, our animals can be healthy. So what are the two nutrients that come from above ground? Well, sunlight. That's onto the leaves. That's why plants have green chlorophyll. There are a number of other photosynthetic pigments. So all of that group of pigments, sunlight is absorbed. What does your plant do with that sunlight? It's got to store that sunlight somehow. And so what your plant has done, what nature figured out, was to take two molecules of carbon dioxide from the atmosphere. And through the process of photosynthesis, it's going to link those two carbon atoms in that carbon dioxide. Well, how do you get rid of that oxygen? So you've got the oxygen that goes back out into the atmosphere, and the two carbons are bound to each other using sunlight energy. We're storing sunlight energy in a carbon-to-carbon bond performed by photosynthesis. And so if your plant needs two carbon sugars, it just takes two carbon dioxides and bonds the two carbons together and blows off this oxygen back into the atmosphere. What if your plant needs a three carbon sugar? Well, then it just grabs another carbon dioxide and sticks it on the chain of two carbons, and now you've got a three carbon chain. This is really not rocket science. This is not difficult to comprehend when we put in these kinds of simple terms. What if you need a six carbon sugar? And so when we're making glucose, when we're making fructose, a five carbon sugar, your plant is just going to keep pulling in more CO2s through the process of photosynthesis and making those sugars. Okay, well, great. Now, can your plant grow on sugar alone? No. So now where is your plant going to ship that sugar to in order to pick up all the other nutrients that your plant requires? Well, it ships it down to the root system, because all the other nutrients, the nitrogen, the phosphorus, the iron, the zinc, the boron, the silica, every other nutrient that your plant requires has to be taken up through that root system. But the plant only takes up certain specific kinds of nutrients. They only take up nitrate or ammonium, depending on what kind of plant you want to grow. That the early annual plants require nitrate, higher concentrations of nitrate than anything else. But if you're dealing with a perennial plant, those plants require ammonium as the predominant form of their nitrogen. And in fact, when we look at trees, when we look at shrubs, they will actually be harmed by nitrate being present in large concentrations in the soil. So how is it that nature has managed to put together a system where perennial plants can control things around their root system, so they're just taking up NH4, that's ammonium is the typical form of nitrogen versus nitrate for our annual plants. So can we use that? If we understand how nature is making ammonium or nitrate that are being selected by your plant, can we do a lot to get rid of weeds? Absolutely. Can we do a lot to make the soil conducive to growing the plant you want to grow? And see, this is just the very beginning of understanding how you make the conditions in your soil such that the plant you want to grow is going to be the one that wins. And then you don't have to worry about weeds. Say goodbye to those herbicides. Say goodbye to tilling. Say goodbye to crawling out into your garden on your hands and knees and pulling weeds. I do not enjoy pulling. Well, it is therapeutic to pull weeds, I suppose, but maybe for only an hour or two a week, no more. So let's learn how Mother Nature makes certain that those weedy species do not grow. So we'll get there through the course of today. So I've been talking a lot about soil, and we need to understand what the difference between soil and dirt actually is. Because we change our soil into dirt when we use pesticides, when we use inorganic fertilizers, when we till too much. We're going to convert that soil into dirt because of the toxic chemical effects of the pesticides, the salt effects of inorganic fertilizers, and the fact that we slice and dice and crush and destroy the organisms in our soil when we till. So as we're converting soil into dirt, or we then need to go back and convert our dirt back into soil. What's the difference between soil and dirt? And so it is summarized in this particular slide. Hans Jenny, who's the father of soil science, said that soil is comprised of the mineral component, but also organic matter and aerobic organisms. When you read Hans Jenny's work, he was very adamant about these had to be aerobic organisms. Anaerobic organisms are the disease causers. They are the pests. They are the problem organisms. Not all of the anaerobic organisms actually attack and kill and harm plants, but they're not helping your plant at all. And they are helping the disease and problem organisms to win in competition with the good guys. So we have soil is comprised of all of those three things. So let's just spend a few minutes defining exactly what I mean by the mineral component. Because if we destroy the organic matter in the soil and we destroy the organisms in our soil, we will turn that soil into dirt, which is just the mineral component. So the sand, the silt, the clay, rocks, pebbles, boulders, the parent material. Everything on this planet is underlaid by parent material. And so as that parent material breaks down into the smaller and smaller and smaller and smaller components, that's what we call rocks and pebbles. And the smallest size fraction, the clay, are the same size as bacteria. The silt particles are the same size as a red blood cell. And sand grains, of course, you can see with your eyeballs. So yeah, pretty small, but you can still see them. And so as your parent material breaks down into each of the smaller components, ultimately what you're going to be left with is sand, silt, and clay. And so that's the mineral component. There is no structure. It is very hard to move water or oxygen or roots through something that is just sand, silt, or clay. So how do we build structure? How do we get those clays aggregated so that oxygen and water can start to move through your soil and your roots will be able to follow? How do we make certain that the sand, silt, and clays get macro aggregates formed? And that doesn't happen magically, much as soil scientists seem to think that it just magically happens in your soil. It doesn't. There is nothing magical about this. Darn. I mean, it would be so much fun if it was magical, right? But it's not. We have to understand what forms that structure. And of course, it is the organisms. So if we don't have the bacteria to build the micro aggregates and the fungi to build the macro aggregates, and the larger creatures, the nematodes, the protozoa, the microarthropods, the spiders, the earthworms in our soil to build structure, then everything's compacted. Just dirt is terribly compacted usually. As soon as it starts raining, your dirt is going to compact, and good luck growing plants. So we have to get the organisms back into the soil so they do the work of building structure instead of us going out and tilling the soil. So we go out, we till the soil, we fluff the top layers of the soil, but we impose a compaction layer at whatever depth the tines or the discs or the chisels, whatever you're pulling through the soil, compacts at the depth that those things reach down to. And so good luck getting your root systems through. They can't grow through those materials. It's anaerobic. Guess where your diseases and pests are? So we have to get these organisms back into the soil in order that they will start to form the structure, and make sure that that structure goes as deep as your roots are going to go. Well, how far down do roots go? We'll get to that hopefully today. So when we're looking at how do you feed microorganisms, we've got to have the organic matter in the soil, the dead plant material from your previous crop, the root systems from your previous crop. That organic matter has to be present in the soil. One of the things that people don't really understand is that when we look at the mineral component in your soil, there is a massive amount of every nutrient your plant could possibly ever need. And there is on the order of 20,000 years worth of these nutrients in every gram of your soil. So why are you putting on inorganic fertilizers? If all those nutrients already exist in the sand, the silt, the clay, the rocks, the pebbles, your parent material, they're there in every gram of that rock material, the crystalline structure. Why are we putting on inorganic fertilizers? Who's been lying to you? Oh, wait a minute. We need a way to get these nutrients that are in your soil back into a plant available form. See, when I was talking about that ammonium and the nitrate, that's the form plants have to have. When we go to the minerals, so let's take a look at this table, the minerals in soil. Sparks, 2003, put together, went out to soil chemistry labs all over the world and got data from the soil chemistry labs and looked at the average concentration of all the nutrients. Now, I've only included the top 20, but let's look through here. Can you find nitrogen? So down here towards the bottom, there's nitrogen. Look at the fact that there is 2,000 milligrams per kilogram of your soil already present in your soil. How many of your plants need more than 2,000 milligrams per kilogram of your soil? That's more than enough to grow any plant you could possibly think about. It's already in your soil. Why are you putting on more? Phosphorus. Those people are in chemical companies saying, oh no, we're running out of phosphate. We're going to have to put massive amounts of money into figuring out how to fix more phosphate so we can keep selling people fertilizers. What? Look at how much is already in your soil. There is no plant on this planet that needs more than this per kilogram. So when you actually think about how far down the root systems of your plants grow, there is no way in God's green earth we need to be putting on inorganic fertilizers. But we got to convert these not plant available forms. These nutrients are in the crystalline structure of the clays, of the silts, of the sand, of the rocks, the pebbles. How does Mother Nature convert all of these nutrients into a plant available form? And oh, surprise, it's biology in your soil that does that. We have to feed those organisms.
Yes.
We have to make certain that the organisms are present and functioning. So we got to feed them, and we have to make sure the organisms are there. So if you're killing your organisms off, then you can't get these nutrients which are in your soil. So who's making money? If you're going out and you're buying those inorganic fertilizers and you're putting more on and more on and more on every year, and most of that is now washing out of your soils and into, because once we lose that life in the soil, there's nothing to hold on to the soluble nutrients. When these things get converted into soluble forms, there's nothing to hold on to them. And so they leach, they wash out of your water into your surface waters into your groundwater, and now you've got to pay money to clean up that water. Or it's going to destroy the Great Barrier Reef. It's going to destroy your ability to drink that water. All around the world, we're seeing water wars starting to happen where people are being killed for control of the source of fresh, clean water that's still drinkable. You would never want to drink the water that's going through an irrigation ditch. You probably wouldn't live long if you did. So how do we clean up our act? How do we, well, get the biology back into the soil? We access these nutrients which are in your soil to begin with. This is what Mother Nature has been doing ever since life started on this planet. Bacteria make the enzymes to pull the nutrients out of the sands, the silts, the clays, the rocks, the pebbles. Fungi, which come along a little bit later than bacteria, they too have the enzymes which break down. Fungi are better at pulling the phosphorus, and converting it into phosphate in their bodies. So the organic forms of all these nutrients, the soluble forms of all these nutrients, are made by the bacteria and fungi making the enzymes to pull those nutrients out of the crystalline structure and into their bodies in the organic structure. And then, how do we make the nutrients that are tied up in your bacteria and fungi available to your plant? Well, Mother Nature came along with a different set of organisms. So when we're looking at bacteria, they have to be eaten by protozoa, bacterial-feeding nematodes. And because the nutrient concentration of all nutrients is so much higher in the bacteria, when the bacteria are eaten by their predators, protozoa and bacterial-feeding nematodes, the excess amount of nutrient is going to be released into the soil in a soluble form, plant available. So how does the plant control all this? Well, let's go back to that root system, and we realize that all these sugars coming down from upstairs, from photosynthesis above ground, those sugars come down into the root system, and the plant is going to release some of those sugars. Some sugars, some compounds that are more conducive for the fungi to grow very rapidly and make their enzymes to pull nutrients into their bodies. Some of them are going to be for feeding bacteria. If the plant needs more silica, what's the plant going to do? It's going to put out the sugars that feed those bacteria or fungi that make the enzymes to pull silica out of the sand, the silt, the rocks, the clays, pull it into the bacterial, fungal biomass, and then here come the protozoa, the nematodes, because the bacteria and fungi, their prey groups are growing. And so here come the predators. They eat the bacteria and fungi, release the nutrients, and the plant is right there saying, thank you, thank you. That's exactly what I needed, and everything in proper balance. Because who's in control of which bacterial and which fungal species are going to grow and make enzymes to pull nutrients in, and who's in control of what kinds of predators are going to be attracted into that root system? It's your plant. Isn't nature clever? It's just astounding that human beings invented inorganic fertilizers, started putting them out because poor Mother Nature didn't know what she was doing. Nature needed some help, and we were going to help nature do her job. When in fact, with those inorganic fertilizers, the constant tillage, the use of pesticides, we were killing the very organisms that do the work the way nature set things up. So we have to stop. We've got to turn this around. We've got to get biology back into the system. So here's the food web. We've been talking about sunlight and CO2 going through photosynthesis. Now we got the sugars going down to that root system, and those exudates coming out of that root system to feed bacteria, building structure, holding nutrients. Nothing leaches out of soil if you've got microorganisms growing in that soil. We hold on to all of those soluble nutrients because the bacteria and the fungi are going to take them up and hold them in their bodies. And most of the bacteria and fungi are growing right around that root system. So high numbers of bacteria and fungi. Protozoa are now moving into that root system. The nematodes are moving in. The fungal feeding nematodes, the bacterial feeding nematodes, eating those groups. Here we are, microarthropods, which eat fungi for the most part. And so they're eaten, releasing the very nutrients that your plant needs to grow. Get that back into your soils, and you don't have to put out inorganic fertilizers. Make sure that things are balanced in your soil correctly so that we select for the plant you want and not for weeds. And you don't have to weed anymore. You don't need herbicides. So getting this biology back into the soil, where are you going to find these organisms? Where do they exist? Where are you going to get them in high enough quantity? Well, we're lucky that all these microorganisms reproduce themselves. And so we don't have to be putting them back into your soils at the exact precise concentration that they're needed. We just have to make sure we put the inoculum in plus the foods to feed them so that they will reproduce and get to the level. The plant will help you make certain that these balances are going to be exactly what your crop plant wants. So we need to help. And what's the best way to figure out whether you've got the organisms in your soil or not? Microscope. It doesn't take more than a day for us to teach you how to use that microscope, teach you what these different organisms look like. And yeah, then you need a little practice after that. You need to be looking at your own soils and getting familiar with what all your critters look like so that you can identify the good guys and you can identify the bad guys. When you start seeing ciliates, then you know something's going wrong. And now what do you need to do to fix the problem? What happened that the problem developed so that you don't allow that to happen ever again? So let these guys do your work for you. Instead of you having to do all the work, put back the organisms that nature has always meant to do the work instead of us. So understanding this biology is critically important. A lot of times people will say, okay, so we need bacteria and fungi, and we need protozoa, these bacterial feeding, fungal feeding nematodes, and the fungal feeding microarthropods. See, we don't really need these guys up here. Oh, no. That's not how Mother Nature works. We need this diversity. We need maximum diversity of everything. So no matter what the temperature or moisture or climate or plant you're putting into the system, we will have the right microorganisms present to do the work that that plant requires. And think about this, that if too many of the predator species are present in your soil and they're happily growing, eating the bacteria and fungi, releasing nutrients, bacteria and fungi are growing and they're building structure and they're taking up nutrients, converting what's in your crystalline form in your sand seals, clays, rocks, and pebbles, etc. All of that work going on. If you get too many of these guys, of the predators in the system, they will eat all of your bacteria and fungi to very low levels. And now your plant's in trouble because no nutrients are being made available. No bacteria, no fungi. It doesn't matter how many exudates your plant puts out. It's not going to be growing the good guys. Probably going to be growing the bad guys. So we have to have something that's going to balance the predators of the bacteria and fungi. We don't want too many. We don't want too little. We want enough. So, Goldilocks principle, not too hot, not too cold, not too wet, not too dry, all of that good stuff. And so how does nature maintain these populations at that, critical balance point? Well, she sends those guys into the system. The predators of the microarthropods, the predators of the nematodes, and they also, both of these groups here will also eat protozoa. It's like, if you run in front of my face, I will eat you. So they keep the three of these in balance. Well, what if we get too many of these guys? Then they're going to overeat these guys, and then there's going to be too many bacteria and too many fungi. It is possible if you don't balance the system. So we want to make certain that there's predators of these guys. So who eats these predators? Well, yeah, there we are. And there's who eats them. Yeah. So who's the top on this food chain? Well, it's a food web, isn't it? There's lots of connections between everything. So it's a food web, not a food chain. Who's the top? We are human beings. We're supposed to be the gardeners of this planet. We're not supposed to be the exploiters. We're not supposed to be the destroyers, because if we destroy it, we destroy ourselves. There's a great book by David Montgomery called Dirt, The Erosion of Civilizations. Every civilization on this planet has destroyed itself by destroying their soil. It took the Roman Empire 2,000 years to destroy their soil to the point they could not grow food to support their own population. How long has it taken us to destroy our soils? Yeah, we're on the cusp, and we've only been doing it for 200 years. So we've speeded it up with bigger equipment, bigger machinery. We've speeded up the process. We need to get that food web back into the soil. So we will suppress the disease-causing organisms. We have to get oxygen back into our soils. We've got to make certain we have the right sets of microorganisms, the aerobic organisms that will compete with, inhibit, and consume the disease and the pest and the problem organisms. And when you do that, you don't need pesticides. Throw it out the window. Well, maybe we keep them just for backups when Mother Nature does something really unusual. And she does do that every once in a while. We need to retain nutrients. So the healthy food web will keep those nutrients in the bacterial and fungal biomass. So you don't have to worry about erosion. Everything's tied, everything's glued, everything's bound. That's what needs to happen in our soils. True soil will do that. Leaching doesn't happen because the microorganisms will take all those soluble nutrients and pull them right back into their biomass. Hold, retain, nothing leaches. We'll clean up water. We don't have to worry about our water sources if our soils are healthy. We're going to make nutrients available at the rates the plant require because it's the plant in control of this process. We don't need inorganic fertilizers. And if you really think about this in the organic world, we don't need organic fertilizers. We may need organic matter as food to feed the bacteria and fungi and keep them going when our crop plants aren't out there growing. But we don't need organic fertilizers, nor do we need inorganic fertilizers. So we have to have bacteria, fungi, protozoa, and nematodes at the very least. We've got to have those groups back into our soil in high concentrations and in high diversity, properly balanced for the plant we want to grow. And we will hopefully get to that part. We've been doing this for an hour here. We've got another half hour to go, so I better talk faster. So we need to decompose toxins. If there was compaction that occurred in your soil, those toxic chemicals that are produced under anaerobic conditions, and I will go through those toxic chemicals with you and explain what's going on. But we need to have things, the microorganisms, aerobic organisms, that will get rid of those toxins and prevent the damage to your plant. Building soil structure. As soon as we build soil structure, we will hold more water. Even dewfall. And when we start looking at the prairies of the United States, where we're in severe drought, California, we've had a drought for the last 5 years. And then all in the course of 1 month, we get 5 years worth of water and rain. Great. Nature does do strange things. So we have to have the biology in the soil to deal with that. So most of our crops are actually growing on the dewfall coming into that soil. We've got to collect and hold and allow to infiltrate into our soils every drop of dew that happens every morning and every evening. And so we can keep growing our plants. But we better have structure in the soil. The organisms better be there to build the structure to allow infiltration so that water moves into the soil and does not evaporate. So we will increase water holding capacity as that structure is built, and that allows oxygen and water to move deeper. And when your water and your oxygen is deeper and deeper into your soil, your roots will grow deeper and deeper. And then you don't have to do anything to baby your plants to try to keep them alive. Put your seeds in, make sure the right biology is there, and let them go. I often say we need to teach everybody how to get this biology back into the soil. So you put your seeds in and you walk away and go fishing for the rest of the summer. You come back and harvest.
Can I
You
just
don't.
interject there? Can I just interject there a little bit, Elaine, just
Sure,
quickly?
sure.
Everything that Elaine's talking about here and in the rest of the slides has been verified 10, 20, 100 times over with work that we've done here in Australia using Elaine's science. I'd just like to reiterate that. This is not just theory. This works in practice as well. So I just wanted to put that in because we know that to be true. We have hundreds of case studies here in Australia to support what Elaine's saying. And as she mentioned before, she's now also in the process of some decent, year 2 or year 3, actually, I think it is.
Yep. Start of year 3.
Exactly. So sorry, Elaine, I just thought I had to pitch in there.
Good point. Yeah, good to say that this is not conjecture. The basis for most of the information that I just went through is in an ecological monograph that was published back in 1985. The very first original paper in soil ecology that put all of this nutrient cycling information, this soil structure information, water holding capacity, was published back in 1985. There's the original paper. And, it's an Ingham et al., 1985. Go looking for it in ecological monographs. If you want to find that reference, go to my website and look in my curriculum vitae that lists all of the publications and find that one. I am not actually the first author on that publication. I'm the third author because I did the work on the bacteria, the fungi, some of the protozoa, a little bit of the experimental design, but it was really my husband's PhD research that he put together and got the publication in ecological monograph. That is very rare for a graduate student to do work of that quality. But you can see that between my husband and I, we have really interesting conversations at dinner time. And I think we scared both of our kids off. They didn't, enough science. I don't want to listen to that anymore. So both of my children are in the world of entertainment. That's what happens when you have two scientists for parent. So for parents. So I've been talking about bacteria, fungi, protozoa, nematodes. I want to make sure that people have a mental image in their mind of what these organisms look like. And, so what's the morphology? And we'll again go over the function of these different groups. But I'm going to show you some good guy, bad guy pictures. So you start to pick up on that particular, what do the bad guys look like? What do the good guys look like? And remember, it's really aerobic, anaerobic conditions that are going to select for these different good versus bad guys. So in this particular picture, total magnification of 400x. So we're not using any really high-powered, highly expensive microscope lenses. These are, the lower end of the range. We don't have to use, transmission electron microscopes or scanning microscopes. Don't need that. These are just simple shadowing light microscopes. And so one of the things we have to train you to do is how do you do the shadowing? Most microscopists are clueless. When it comes to that, it's a very simple procedure, but it does take, it takes about 10 seconds of training you to go from just simple light microscopy to shadowing light microscopy. So it's not really very tricky, but you have to realize what you do exactly. So we're looking at this picture, shadowed light microscopy, total mag of 400x, total magnification. And in this picture, can you find the bacteria? Can you find aggregates that have been built by the glue that the bacteria make? Can you find the root that's in this picture? And can you find the ciliate? The ciliate is the bad guy. And there's a couple other pieces of evidence when you look along that root system that we are dealing with a system that's gone anaerobic. There's evidence here that says in the past, this has been a very healthy soil, but something's happened and happened fairly recently, such that your plants are dying. So we went looking through this soil going, okay, what's causing the problem? What's happened? So we're looking at the individual bacteria. So if you follow the little pointer, you can see each one of these little bacteria. You can see we basically have one size of bacteria. They're all round, cocci. Some of them look a little bit bigger because they're more out of focus than other ones. So where are the bacteria actually in focus? It's in here in these aggregates. Bacteria make glue. They make copious amounts of glue because the bacteria don't want to be washed away from their food source. So they're going to glue themselves onto the surfaces of these aggregates. They are going to build those aggregates. So all those little grainy things in there, those are all bacteria. This particular aggregate is glued to the surface of this root system. Inside there, you can see that the bacteria and fungi have been doing their job for a while. They have been condensing plant material down into the fulvic acids, that light tan honey-colored material, and the humic acids. So right there, you can tell whether you've had good microbiological processes going on in the past by looking at the color of the aggregates. If your aggregates have no color, be kind of worried. Those aggregates are not water stable. Aggregates like this should be water stable. They don't break up when you mix it in water. You can see that there are rod-shaped bacteria, but again, there's just one kind of rod-shaped bacteria, which scares me a little. The diversity of the bacteria are not what they should be. So I'm already going, got a problem in here. Here's the root. And so you can see multicellular, lots of individual cells in there. And multicellular materials, well, root systems, if they're in uniform diameter all the way along, typically roots will be uniform diameter. And as you go along the surface of the root, you're going to have to kind of look through this layer of aggregate material. Ooh, there's some good humic acid in there, right? It is pretty much uniform diameter along here, but as we get to right here, notice the divot. Uh-oh. There's a disease-causing fungus growing right here and happily decomposing your root system. So the conditions in the soil are now allowing the disease causers, the problem organisms, to win. Where are good guys that should have been preventing that anaerobic disease-causing organism from being able to grow? Almost all the human pathogens, almost all the bacterial pathogens of plants, and the fungal pathogens, disease causers, require reduced oxygen conditions. As soon as that oxygen starts dropping too low, our good guys are going to stop growing. They're going to stop being able to compete with, inhibit, and consume the bad guys, and the bad guys start to grow and say goodbye to your root system. Your roots can't tolerate that. So there's a second factor that's like, wow, this was good soil once upon a time, but it's not there anymore. And the final kind of nail in the coffin is a ciliate. Ciliates are protozoa, so they are eating bacteria, and you can see all the bacteria inside that organism. It's about 50,000 protozoa and bacteria a day. And you can see the little cilia, and that's why we call them ciliates. They have these little hairs, sometimes over all of their body, sometimes over just parts of their body. But any place you see multiple little hairs like this, little cilia, then your heart should kind of, just, oh no, oh no. We're starting to go anaerobic. And when you start to see these things, you've got to start thinking immediately, what can I do to get the bacteria and fungi back in here, to get them to build the structure to allow the oxygen to move back through this soil? What am I going to do? What are the processes? So we've got to figure those things out. What happened that things are going bad in here and the diseases are winning? What do we have to do to bring the proper biology back and build structure? Come on, guys, get back to work. Why did you stop working? What did we do? So learning this kind of information by using a microscope, very important, very critical. And when you master doing the microscope, it takes you 5 minutes to prepare a slide. You take your sample from the field, you mix it up in your plastic bag, you get your quarter teaspoon or your 1 gram measure, and take a gram out, put that into a test tube, and then add 4 milliliters of water. Shake it for 30 seconds. Take a drop out with a little pipette or an eyedropper. Put a drop on your microscope slide. Cover slip on top of that. There you are. You put your sample under on the stage in your microscope, and now you're looking. Here we are. Here's another view. So what are all these strands? See all those threads? We already know what these aggregates are, right? And you can see that we've got, well, a little bit of humic acid in here, but nothing like that last sample, huh? Lots of clear, colorless little aggregates. So this is not as good as soil from that point of view. This is probably a soil that's just being built, just coming along. But these strands here are fungi. And so we can measure the length of the fungi, we can measure their diameter, we can start to tell how many different species of fungi, because there's a different fungus, there's a different fungus, there's a different fungus. How can we tell if these are good guys or bad guys? Wide diameter, anything wider than 3 micrometers. So find your littlest bacterium up here. Bacteria are 1 micrometer in diameter. So if you go looking for the little guys, how many of those little guys can you put in here? About 3. So this is 3 micrometers wide. You've got built-in scale for yourself when you're looking through the microscope. And so good guy fungus, because it's wide diameter, it's dark in color. There's another point, that any colored fungal hypha is good. Yes, good stuff. It's the clear, colorless stuff. Yeah, but see how wide diameter that is? Okay, it's wide diameter, but it's clear color. So you can start ranking your fungi. This is the best fungus in the group. That's still a pretty good fungus. How about this guy? Do you see any color in there? Yeah. How wide diameter? Okay, it's maybe it's 3. But here's the problem. If you focus on this sharply, this is only 2 micrometers in diameter. It's clear, it's colorless. There are no septa. It's a disease-causing fungus. But is that disease-causing fungus going to be able to grow and outcompete the beneficial, all that beneficial fungus in here? No, no way in God's green earth. If we are dealing with aerobic soil. So good guys in here that will be able to outcompete, inhibit, and deal with any disease causer, so we don't have to worry about it. I need to get going faster, so I'm going to start going faster. Another fungal hypha. And so septa inside, if you see these cross walls at uniform distances, that's a really good indicator that this is a beneficial fungus. See how wide diameter it is as well? Yeah, good stuff. Kind of a funny color, kind of the brown bluish, because the background is bluish. It's giving the fungal hypha a bluish cast, but it's clearly a much darker brown color. So it's colored, it's got septa, wide diameter. It has to be a beneficial fungus. Looking at mycorrhizal fungi, we can look for mycorrhizal fungi inside root systems. This is a root system that's been illuminated with ultraviolet light. The arbuscules of the vesicular, arbuscular, or endomycorrhizal fungus is fluorescing, autofluoresce. And so you can see where we've got colonization inside the root system and where we don't. Is this adequate colonization for the plant you're trying to grow? So we can easily look at mycorrhizal fungi. Here's ectomycorrhizal fungi, forming a sheath on the outside of the root system, and then the hyphae grow out. And so with any kind of mycorrhizal fungus, the plant lures that mycorrhizal fungus to the root system, forms the relationship with the fungus, and then the fungus is told by the plant what nutrients to go out, and collect. So the fungus makes the enzymes using the sugars that the plant is feeding it from the root system. Fungus goes out, collects the nutrients, pipes them right on back to the plant, and gives the plant what it wants. Both organisms grow better. It's a mutualism. So most of the plants that we grow as vegetables need to be mycorrhizal, except for the brassicas, the mustards, the cole, and the kale crops. Those we don't want to be mycorrhizal. So in those soils, we've got to prevent mycorrhizal colonization on the roots of the plants. And so we get the actinobacteria into the soil in high concentrations, and the actinobacteria will suppress the ability of that mycorrhizal fungi to colonize the root systems of those plants. So, understanding biology, we can start selecting for exactly what we want. You can see fungal hyphae with the naked eye when they form these rhizomorphs, where lots and lots and lots of strands are in here so that you can see them with your eye. If you picked up this piece of organic matter, you would pull all the rest of this organic matter with you, kind of like a wind chime or something, because those fungal strands are so strong. They will hold all of those bits and pieces together. So you can see where fungi form macro aggregates that will allow even better water holding capacity within your soil. Now going on to the predators. Here's a flagellate. There's a flagellum, and it has another flagellum out there, which is why we call them flagellates. They eat bacteria. And so you can see a bacterium in its body. You can see a vacuole. It's about to poop. Bacteria poop contains all those soluble nutrients that your plant wants. And so we want these guys to be very close to the root system. They are attracted by the bacteria and fungi that your plant is feeding with the exudates. Now, when you look at this soil, look at how little aggregation, and there's absolutely no humic or fulvic. So not a very well structured soil. I'd be a little bit worried about that soil because it just doesn't have much structure. Do we have compaction problems in that soil? Well, but at least we've got bacteria, lots of bacteria, lots of bacteria. And at least we've got some nutrient cycling going on because we can see that flagellate. Nematodes. So we identify nematodes based on the mouth shape. So this bacterial feeder is going to be consuming 10,000, 20,000 bacteria a day to keep itself alive. When it eats those bacteria, it's now going to release the excess nutrients. C to N ratio of that nematode is so much wider than the C to N ratio of the bacterium that all those excess nitrogens are going to be released. Well, same holds for magnesium and calcium and sodium and potassium and phosphorus and every nutrient that you want to talk about. So very important to have these predators. This is a fungal feeding nematode, and what we want to look for in its mouth is this spear that it uses to puncture the cell wall of the fungal hypha, sucks out the internal contents. Excess nutrients pooped out of the nematode in a plant available form. Predatory nematode. Again, mouth parts. We're looking at this great big tooth, great big mouth. And so a predatory nematode is going to grab the other nematode and just chomp, pull them in, chomp, 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 grab another mouthful, pull it in, chomp, 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 grab another mouthful. Isn't soil a nice place? All these who eats who situations. Root feeding nematodes, you really want to be able to identify these guys. And the simple way to identify them is to find the spear in the mouth and look for the knobs. Only root feeding nematodes have great big knobs like this. And they're usually pretty easy to identify. If you've got nematodes thrashing around and they won't stop wiggling, you just take your microscope slide off your microscope and run that microscope slide through a flame about 10 to maybe 20 times. And that'll slow the nematode right down so you can see whether you're dealing with a root-feeding nematode, a bacterial-feeding nematode, a fungal-feeding nematode, or a predatory nematode. So organisms do all the work in the soil. They are what do all those important things. All the reasons that we use pesticides and inorganic fertilizers, the green revolution that we were so proud of, only occurred because we had already destroyed all the biology in the soil. We had turned our soils into dirt. And the only place that the green revolution works is in dirt. If you have the biology in the soil and you put an inorganic fertilizer on, you're going to kill those organisms. So now you put the fertilizer on, the plant responds, whereas it did not respond when you had a good, healthy set of organisms in the soil. So if you're putting an inorganic fertilizer on and your plant responds, you know there's something wrong with your soil. And you need to get in there and put the right biology back into the system so that if you put an inorganic fertilizer on, you should get no response. And then because you killed your organisms with that inorganic fertilizer because you were testing to see whether you needed the inorganic fertilizer, now you do need that. Now you do need that replenishment of the biology in your soil. So hopefully everyone's getting this pretty well under their belts. How far down do roots go? Well, they go as far as the roots can go based on the physiology of the plant. Most of us were taught that root systems of grasses, for example, don't go down any more than maybe 1/8 of an inch. Talk to golf course superintendents. They all had that little piece of mythology in their brains that the reason the root systems of their plants only went 1/8 of an inch down is because you cut the grass to 1/8 of an inch. The root systems only went down as much as the above ground part went up. Absolutely not true. When we have good biology, when the soil is not compacted, the roots will go down as deep as they possibly can. So these grass seeds were planted in July, allowed to grow until November 6th, and we punched a core, pulled that core out. Yes, it took a big crane to do it. Washed the soil off of that root system. And this is a 6.5-foot tall person holding this core of plant material. And that root system on that plant, even though we had mowed this grass to 1/2 inch, approximately 3 days before this picture was taken, before we removed the core. And you can see that the grass has started to regrow. We mowed to 1/2 inch. So either mowing does not cause the roots to slough and fall off, or you're trying to tell me that the root system on this grass, when we mowed it to 1/2 inch tall, sloughed off, and then they regrew. In a 3-day period of time, they went from 1/2 inch to 4.5 feet, over 1 meter in 3 days. Yeah, pretty ridiculous, huh? You don't cause the roots to slough off. Those roots are established. The plant has spent the carbon. It doesn't keep turning the carbon in those root systems over and over and over that rapidly. They're stable structures. Your plant needs it to pull in nutrients to feed itself when the above ground part of your plant starts growing again. There's no way it's going to slough those roots. Now, if you mowed this and a big set of exudates go out of that root system, bacteria and fungi start growing, they start doing the work the plant requires, and just at the time the above ground part of this plant grows, that's when here come the protozoa and nematodes and big burst of plant available nutrients given to your grass plants. But what if you come in and you mow this again, or you let animals graze this again, before the above ground part of that plant material is recovered, and your plant has replenished the exudate pool, the sugars have not been replenished yet, and you come in and you mow it or you graze it again, too soon, you're going to stress your plant. Now your plant's starting to go, oh, I don't have enough exudates to feed the bacteria and fungi and get the protozoa and nematodes to come in here and give me that big flush of nutrients that I need in order to grow the top part of my plant. What if you come in and graze this too soon yet again? See how every time you do that too soon, you don't allow the plant to recover? it's going to stress your plant more and more and more to the point where it will start to slough its root system. So be aware that you really have to understand what's going on below ground. Now, a picture from the United States, Great Plains of the United States. People went out into the Great Plains. They dug down into the soil 40 feet, took a fire engine and washed that surface off, put plate glass, put tracing paper over that, and traced the root systems of each one of these plants. Then they took those tracings and lined them up in this photograph, took a picture. How far down do roots of grasses go? Well, it depends on the grass, doesn't it? The root systems on these highly productive plant species are down at 25 feet. Little less productive, well, they're maybe 15 feet, they're maybe 10 feet. The least productive grasses, yeah, look at that guy. That's a perennial weedy grass species. We don't want those in our soils. How do you select against these guys? They're not mycorrhizal. So get mycorrhizal fungi back in here, and all your other good guys, more productive plants, will exit the not productive plants. Cheat grass. Can you find cheat grass in this picture? Yeah, no root system to speak of at all. Very early successional plant species, comes in, grows very rapidly, sucks up nutrients and water, produces seeds, and goes on about its life cycle. Very short, very early successional. It doesn't need a lot of root system. It puts all its effort into making seeds. So succession, we have to start understanding the effects of disturbances. So while you don't disturb a system, or nature doesn't disturb a system, going to develop these nice forest systems, hold all the nutrients, no runoff, nothing being lost from the system to speak of. Along comes a fire, along comes a herd of buffalo, along comes a herd of kangaroo, and starts to disturb that system, a flood, a fire, something like that. And now the plants have got to come back from that disturbance, and they can't go immediately to a beautiful forest system. So here's where you start losing more soil. We don't have the biology, we don't have the plant species, way more early successional plant species. What if we come in and we are tilling and we are disturbing that soil and slicing and dicing and crushing? Really high levels of soil loss. So we need to understand the successional process. What we find is if you're dealing with bare soil, the only thing in bare soil is going to be a little bit of bacteria. And you can't form structure, you can't hold on to your nutrients. So you say this is a really poor soil because every time it rains, all of your soluble nutrients are leaching out of that soil. Oh, this is a poor soil. No, it's not. It's lacking biology. It's lacking the organic matter to grow the organisms to start building soil structure, start building that soil. As we start to get the weedy species coming in, now we at least have some fungi. We're starting to build better structure. We're holding on to more of the nutrients. As here are our grassland species coming in, early successional, but way more fungi. As we start to get to highly productive agricultural systems, highly productive grasslands, equal biomass of fungi to bacteria. As we move over into the fungal dominated, more fungal than bacterial, we're going to select for the growth of perennial plants. So shrubs, trees, evergreen, extremely fungal. In some of the most productive ecosystems on this planet, old growth forests, the amount of fungi in that soil would be 70,000 micrograms of fungal biomass per gram of soil. 75% of the volume of 1 cubic centimeter of that soil is occupied by fungi. So when somebody says that soil doesn't contain more than 5% of its volume in the microorganisms, they have never looked at anything except chemical agricultural soil. They would never say that
We
if they
might
had
just
paid attention to other ecosystems.
we might
As
just
we go from that early successional, very nitrate-dominated systems, as we start to improve that fungal biomass, we get more and more ammonium. Weeds require strictly nitrate. So the more we can get that fungal biomass come along in our soils, the fewer and fewer weeds you're going to get. Most of our vegetables, most of the crops we care about, grow in this part of succession. So get rid of your weeds by getting rid of the nitrate. Oh, you're putting urea out? Guess what urea almost instantaneously turns into? Yeah, nitrate. So succession. Now, this is what nature does. So how come nature hasn't gotten everything to the stage of old growth forest and kept it there? Because we have disturbance. All these different things that can push us somewhere backwards, and then nature starts to regrow that soil, and then another disturbance and nature starts to regrow, and then another disturbance and nature starts to and then another, and then another. It's kind of a yin and yang, isn't it? Back and forth, back and forth. How many times have your soils been old growth forest? How many times have they been bare soil over the existence of this planet? Yeah, so many times that we can't even count them. So where are your soils in terms of matching up with the sets of organisms you want to grow?
Can you hear me, Elaine?
So compaction, I had promised that I was going to talk about that, but we're out of time. Maybe just really quickly, when we go from aerobic conditions to anaerobic conditions, it's not the organisms, it's not the organic matter that's going to cause you a problem. As you go anaerobic, all of your soluble inorganic nitrogen compounds will be converted into ammonia, and that is a gas. You go anaerobic, you are losing your nutrients. You will not be able to grow the plants you want to grow. Sulfur. The problem is not with the organic material, it's with the soluble inorganic sulfur forms under anaerobic conditions are converted into a gas, hydrogen sulfide, and it stinks. Most of you can tell when something starts going anaerobic because you smell ammonia, you smell hydrogen sulfide, rotten eggs. Under anaerobic conditions, phosphate, PO4, is going to be blown off as phosphine gas. So think of all of those sulfur soils that you've got in Australia. There are no nutrients left in those soils because they've all blown off as gases. How can you possibly grow a decent set of crops in there? And only under anaerobic conditions are we going to produce these really nasty acids. So as you drop in oxygen, these organisms that produce these organic acids in high concentration and very low pH, that's the only time they're going to be produced. So if you've got an acidic soil, it's anaerobic. It has been anaerobic for some time, potentially. And how are you going to fix them? You're going to have to get organic matter back into the soil. You're going to have to be careful that you don't have interesting acid-base reactions happening in some of your soils. So you have to add organic matter.
Oh, not sure what happened there, but it looks like Elaine has temporarily left us. Probably a bad connection. If Elaine can't come back to join us, I'd like to thank everybody for attending this webinar. One or two minor software glitches, but we'll sort those out for future ones. All of the links for online courses, etc., I will place in the description of the replay on our YouTube channel. Oh, here's Elaine. She's back, I think. You're back.
I started that. I got my microphone on. It's probably a good place to stop.
I don't know. I'm not sure what happened there, Elaine. I was just explaining to people that sometimes these things happen. But there are just quickly one or two questions. Belinda has asked, where did this myth of root sloughing originate from? I think you were talking earlier on. So that's one
There,
question.
there were researchers both in the United States as well as in Australia that came up with that explanation for why roots are really short on grasses. That when you're seeing grazing that grazes the above ground part of the plant to 2 to 3 inches, and then that's why the root systems are only down at 2 to 3 inches. Golf courses, when you're cutting the grass. And so it was just an empirical observation. And so they came up with the idea that the roots sloughed. They had no evidence of dead root organic matter in the soil being increased. They just came up with that explanation. And I think, a lot of people, Christine Jones was one of the people who really promoted that idea. And last year when I was at Quivira conference, I was there and Christine was the keynote speaker. And I'm in the audience going, Christine, no. And she said, at that point, she said, yep, it has been proven that I was incorrect. She said
Oh,
that
nice.
root systems do not slough just because you cut the top part of the plant or because you've got grazing. So enough times she's seen where grazing occurs and the root system doesn't slough as long as you have healthy biology in the soil.
Okay.
You stress your plants, then you're going to be in trouble.
Just another, perhaps one or two quick questions. There is a question that somebody's asked, not sure who, but is it possible to obtain results of vermicast soil amendment for broadacre farming in Australia? Are you aware of any actual studies that have been done on that subject?
Of using worm compost
Yeah.
to as the source of biology? Absolutely, yes.
Yeah.
There's quite a number of them. I was just looking at some of that research today. I think there's over 100,000 papers in the scientific literature showing that worm compost biology is every bit as good and can increase yields in exactly the same way that thermal compost does. The important point is to get that biology growing. And earthworms, of course, they're born with a set of microorganisms in the back end of their bodies, in the back chamber, that they get from mom. They're with them at birth. And those are some of the best microorganisms in soil for suppressing diseases, for cycling nutrients. And it's a huge diversity, not only bacteria, but fungi, protozoa, and nematodes. So the full food web is in that earthworm. Of course, you got to eat them, give them a variety of foods to eat. And another one of those misconceptions that's out there is that earthworms will eat organic matter. No, they don't have the enzymes to break down plant material. What they do is crush the bacteria and fungi, protozoa and nematodes, and the juice that spills out, that's what the earthworm takes up.
Okay, well, that's good. I mean, there's probably 100 questions that people have got, and some people have had a little bit of trouble viewing the webinar today, but I will post all of the online links, etc., in the description part of this replay. So people that have missed it or haven't been able to see it properly will be able to see it in the replay. We'll also be announcing potential dates for Elaine to come out to Australia. She hasn't been here in a long time. She misses the sunny weather and the Aussie people, which is good. And I'm sure we will have more of these as time goes on. In fact, we may plan a simple question and answer webinar, for example, Elaine, or something similar. So, big thank you to you, Dr. Elaine Ingham. Sorry?
I think you can see that I ran out of time. So I didn't get through to the examples and the data. So we're just going to have to do this again. That's all there is to it. So I can finish this webinar. I can finish the talk that I was going to give and have more time for questions and answers. We meant to have a good 15 minutes of questions and answers. So I apologize for not getting finished.
No need to apologize. Everybody can tell fairly easily how passionate you are about this subject and how passionate you have been for many years. And that's been contagious with a lot of people that have become involved with the software web. So thanks, everybody. We'd better sign off now. Thank you to Elaine, and speak to you all soon. So thanks, Elaine.
Yep.
Log off now.
Thanks for inviting me. Yep.
No problem.
Take care.
Thanks. You too. Bye-bye.
Ciao.